Welcome to the Nightly Show. I'm Jean Splicer. Tonight's special report, is gene editing appropriate for conservation use? Tonight we're going to take a look at clustered, regularly interspaced short palindromic repeats, or CRISPR as it's better known. This is the newest innovation in gene editing technology being used in conservation to help remove invasive species from islands across the globe with minor impacts on the environment surrounding them. The CRISPR system uses the Cas9 protein complex, which contains a piece of guide RNA that contains a target sequence that is complementary to the sequence in the gene being targeted. The Cas9 complex moves along the DNA until the target sequence of RNA finds its complementary match on the DNA strands. The complex then cuts out the targeted piece of DNA. Once the targeted sequence is removed, two things can happen. If donor DNA is added to the solution, then it can be incorporated into the cell's DNA, causing a knock-in effect. If no donor DNA is present, then the DNA will just heal itself without the target sequence. This breaks the targeted gene so that it can no longer function as intended and is called a knockout. This technology can be used to insert genes into invasive populations that do anything from making offspring more likely to be male, therefore reducing the breeding population over time, or it can even insert a disease into the population that will be spread through gene drive to drive the invasive species to extinction in the targeted area. There's a lot of controversy over the use of this technology, so let's hear from some experts and see what they have to say about CRISPR.
And I would just like to mention some of the pros of using gene drives in CRISPR technology. First of all, they don't use any chemicals or pesticides, thus making it much more eco-friendly, and no other animals on the island or other habitats would be harmed. Gene drives also occur naturally, so we would just be increasing the amount of gene drives that occur, and we would not be doing any damage to the already occurring environment. Gene drives can be used to reduce infectious diseases like malaria and other things spread by insects and such other pests. This would be very useful to use in underdeveloped countries, making the death rate much, much smaller. Gene drives can also be used to support conservation work and thus reducing the amount of endangered animals found throughout the world. The CRISPR technology is very, very precise. It deletes a single gene and you can select any of them. And it's also very versatile and simple to use. You can use it on just about any species, and it's pretty much anyone with a scientific background would be able to use it.
Sure, that was very enlightening. Now, let's hear from an activist that is very much against CRISPR.
Since CRISPR is spread through sexual reproduction, there's no guarantee that it will stay within one species, like this rat, for example. Since the rat is related to this chipmunk, it could jump species and end up breeding with this chipmunk, causing the female chipmunk to only reproduce male babies, eliminating out the chipmunk species. Because there's nothing like it in the environment, there's nothing to compare it to. Another problem with using CRISPR is that they want to use it in agriculture to make crops like the corn and rice grow better and become resistant to bugs. They want to alter the corn itself, or they may end up altering the dandelion, a weed, and causing it to become more susceptible to Roundup, a weed killer, but it may backfire and cause it to become stronger, and weeds such as this or the foxtail will overtake your corn, or in this case, the coneflower, which is native to Kansas, killing out the plants that you wanted to keep alive. And then worst case scenario is that governments may get their hands on it, and they may want to use it to release mosquitoes or other bugs that will eat the crops of a country we're at war with or having problems with, and other toxic things, and kill out their agriculture, and it could attack their animals. Or they may release mosquitoes that are infected with Zika or malaria and let it go on the people, killing out the people.
Now, let's go to Dr. Enzyme and see what else CRISPR has in store.
Hi, I am Dr. Enzyme and I'm breaking you down. And today I would like to talk about CRISPR. It is a cheap and faster way to modify plants to be disease and bug resistant. It has been tested on mice to edit out disease, and it hopefully can use on people in the future.
Thanks, Dr. Enzyme. Now, we have one more speaker that has a few words to put in on this subject, Dr. Leafy.
There is a genuine possibility of mutation of target to continue and may increase every generation. There is a risk of transferring modified sequence and passing the negative trait into our food chain. What about effect of having dead invasive species to our environment? Using CRISP to control invasive species? Kidding me? What if they go extinct? What if Osama bin Laden uses CRISP against our nation? Large genome may contain multiple DNA sequences identical to target DNA sequence. CRISP may cleave these unintended DNA, causing risky mutation.
Wow, Dr. Leafy, you seem to be very passionate about this subject.
Disappearance of whole population targeted by gene drive may carry unknown consequences to ecosystem equilibrium.
Thanks, Dr. Leafy. That was energetic. Well, that's all the time we have for tonight, so I'm going to leave it to you at home to decide, CRISPR, should it be used in invasive species management, or is meddling in the genome too much?